freshmen. We are going to be doing our Macromolecules Lab today, and we're going to be testing 4 unknown solutions. You guys should have read that we are zoo nutritionists, and some of the food got mixed up that are supposed to be fed to the carnivores, omnivores, and herbivores. We got to feed them the right food, so we've got to figure out which macromolecules are present in each unknown. Then we can decide which food goes to which animal. So we're going to go ahead and get started, and the first thing I want you to draw your attention to is on page 65 is a little data table. That's where we're going to record our data, and Mr. Kesey's going to come around and show you. I'm going to point out a few things. In our data table, this is a digital version of what you see on page 65. I want you guys to add in some information. You'll notice it says iodine test. That's testing for long chain carbohydrates, also known as starches. The Biret's test is testing for the macromolecule protein. The brown paper test is testing for the macromolecule lipid. And then finally, the Benedict's test is testing for the short chain carbohydrate sugar. So if you haven't done so, pause the video and go ahead and add in which test each or which macromolecule is being tested for based on the testing name. Okay, so we're going to start with the first test, and that's going to be the brown paper test, and we're going to go to see if we can find out if lipids are present in unknown 1, 2, 3, and 4. So what I did is I went ahead and took my pipette and I sampled each unknown, and I put a little drop onto this brown paper bag or piece of brown paper. And what we're going to do is we're going to let this dry, and you can see right now the samples, unknown samples 1, 2, 3, and 4 have been put on there. And we're going to let that sit for probably about 5-10 minutes. And once it's all dry, if the unknown had a lipid in it, when we hold it up to the light, it should be transparent. And so while we're doing that, the other tests, Mr. Ardente is going to hold on to this. It may be a little bit heavy for him, but I think he can handle it.
It would kind of look like, you know, when you get a bag of fast food or something where the bottom of the bag gets all that grease and fat on there. So that's kind of what you're looking for with this test.
Yes.
Simple but effective.
Very good. Okay, good point, Mr. Ardente. Let's go on to the next test. The next test is the carbohydrate test, and we are going to use iodine for this one. So we got our 4 unknowns in there. You'll notice they're labeled 1, 2, 3, and 4. I don't know if any of these or which ones contain starches, which are long chain carbohydrates, but that's where the iodine comes in. Iodine it will change color in the presence of starches. So I'm going to go ahead and add in my iodine, and we'll add in 3 drops to each. So to unknown number 1, 2, 3. 1, 2, 3. We'll add in a little bit more there. 1, 2, 3. And then finally 1, 2, 3. All right, so we added in our iodine, and we'll look to see if there's a color change. If there's a color change from yellow to blue or dark purple, then we know that there is starches present in there. So we're kind of sitting around here. I'm going to let that mix a little bit. Looks still like a yellowish orange to me. Ooh, looks like we have a positive there for unknown number 2. We'll come back to that, but it looks like tentatively that might have some carbohydrate starches in it. Unknown number 3 still looks a little bit yellowish orangish to me, as does unknown number 4. So we'll let those sit for a little bit just to be sure, and we'll come back before we record our data. All right, let's go to test number 4. Test number 4 is the Biret's test, and this tests for the presence of proteins. And I'm going to go ahead and show that our test tubes are labeled with our unknowns. And I'm going to go ahead and add in our Biret's indicator. So again, this will show a color change if proteins are present. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And 1, 2, 3, 4, 5. I think we got it. All right, I'm going to go ahead and mix these. And again, we're looking for a color changed. And if there is a color change, if the color turns to, bear with me here, purple, then we know protein is present. So far it looks like, just initially speaking, there might be protein present in unknown 2, and there might be protein present in unknown 3, but we're going to let that sit for a few minutes before we record our data. All right, let's go to test number 4. This is the final test, and we can see we have unknowns 1 through 4 right here, and this is the Benedict's test. Benedict's tests for carbohydrates as well, but not long-chain carbohydrates, where this one tests for sugars, and sugars are kind of the simple ones. And so we will go ahead and add in our Benedict's, and we are going to add several drops to this one, and we'll again look for a color change to like orangish, greenish, yellowish. Right now it's blue. And this one's got to involve some heating, so we're going to have to wait for the results until after it's been heated for about 10 minutes. All right, so you can see we have a hot plate, and this is heating water. I'm going to go ahead and put these in, and they will sit in here. There we go. Put this in here, get all our samples in there. And we'll have to await the results on that. We'll go back to our lipid test. Mr. Ardente, are those papers dry yet, or are they still kind of wet? Yep. All right, we'll hold them up to the light. Mr. Kedzie, if you want to come back over here, and we are looking to see if they are transparent. And it appears that there are definitely some positive and negative test results. So we'll go ahead and show you the data. And again, the ones that are transparent have lipids in them. So are there any unknowns that appear to have lipids in them? All right. Okay.
I would say 1 and 3.
1 and 3 appear to be transparent, and 2 and 4 appear not to have lipids in them. So you should be thinking about which one of these unknowns would be fed to herbivores and which one of these unknowns would be fed to carnivores based on the results that we see here. Okay, guys, the result is in for our iodine test, and remember, iodine is testing for starches, long-chain carbohydrates, and we can see that there is a positive and that there are 3 negatives. So you should record your data in your data table based on what you see here.
Put a plus for a positive test and a minus sign for a negative test.
Good point. I'll reiterate what Mr. Kedzie said. Again, a plus for a positive and a minus for a negative. All right. So one of these unknowns is positive for starches, which is long-chain carbohydrates, and 3 of them are negative for starches. Okay. All right, for the Barrett test, we can see that we have some, remember Barrett is testing for proteins. 2 of these are testing positive for proteins. That's when you see that purple color. And 2 of these are testing negative for proteins, so not in the unknown. All right. Okay. And then finally, our last test, the Benedict's test. This tests for carbohydrates as well, but simple carbs, namely sugars, which are just not long chain. And we can see that when you see an orangish color, that tests positive for sugars, and that if you don't see orange, that would test negative for sugars. So go ahead and record your data for this one as well. So now to confirm the results, we'll go ahead and compare your data table to the data table that we have recorded here. Remember, we did not do unknowns after 4, and we'll go ahead, and that's how your data table should look. Now, on the next page, page 66, you will be asked to analyze this data. So let's go ahead and take a look at the analysis questions. And I'll come around here. Mr. Kedzie will kind of zoom in on this. So you have your data recorded here. So you've seen that already. So which unknowns have which macromolecules? We're going to go over here to this page. And we'll notice there's a question on data analysis. And it says, using your test results, put an X in the box to indicate which macromolecules are present in the unknown solutions. Be careful not to just copy down the chart as is. They are not in the same order of the above. So what I want you to notice is we have unknown solutions 1, 2, 3, and 4. We can ignore what's beyond that. And here you have the name of the macromolecule across the top. This order is not the same order as what we see here. So you really have to be careful about where you enter your X's for unknowns 1, 2, 3, and 4. All right, guys, good luck, and we'll talk later.